Hi everyone. Many of the students emailed me concerned about what does NHE4 has to do with metabolic acidosis. And so it's not that important for the exam. I know it's one of the objectives, but I'm going to explain it to you, but please don't spend too much time on it. I already answered many of the students, and also we answered it in the lecture, but let me here. Let me summarize what's going on with NHE4. First, to understand at the thick ascending limb, we do have reabsorption that takes place of the sodium, the potassium, and the chloride through the NKCC. And so this is the NKCC or NKCC2 or NK2Cl, anyways. And so we agreed that the potassium here can be substituted with ammonium. So essentially, NH4 plus can come in, and also they can come in from the RUNC, also with the concentration gradient, because you have more NH4 outside than inside the cell, and also the electronegativity inside the cell will help you bring more NH4. So one way or the other, now we have NH4. The question that we have, or you guys have in mind, is this transporter here, which is a co-transporter. It's called NHE4, sodium hydrogen exchanger. It's antiport. I'm sorry, I said co-transporter. But the sodium here is following its concentration gradient because it's always the sodium is higher outside the cell than inside the cell. Later on, the sodium, we can get rid of it with the sodium-potassium pump. That's not a problem. And then the ammonium that we built inside the cell, it's gonna go outside, and that depends. It depends on whether it's going with the concentration gradient or against. But even if there is no concentration gradient for the NH4, just the fact that the sodium is coming in, it's gonna bring the NH4 to the opposite side regardless. And so now we have NH4 plus. If you remember, I told you that we also have here the MBCN1 channel that will take bicarbonate that we made here by the carbonic anhydrase at the TAL. And so you're throwing here both sodium, so you have a co-transporter for sodium, and also HCO3 negative charge. And so they're very close to each other. It's essentially the same cell, so you can expect this HCO3 to impact that loose hydrogen ion that is associated with NH3. So that will result in the hydrogen ion being captured by the HCO3, and this will be dissociated into carbon dioxide and water, and so it's not really impacting your acid-base balance anymore because you can breathe the carbon dioxide out. But it's leaving behind, once you take out a hydrogen ion from the NH4 plus, then it's leaving behind NH3 and ammonia. And that ammonia, if you remember, this is the thick ascending limb here, but over here is the collecting duct. And we agreed that the collecting duct, we do have transporters that will take NH3 and will secrete it, not only by diffusion. We have actual transporters that will take NH3 into the filtrate. And if you remember, we call those RHBG and RHCG. That will force the NH3 to go into the collecting duct. And guess what we have also in the collecting duct? We have type A intercalated cells that are secreting to you hydrogen ions. So this hydrogen ion is looking for something to buffer it. And if you are relying on the molecules that have been already in the filtrate, they're already saturated with hydrogen ion. You need fresh NH3 to capture the hydrogen ion that will be secreted with the intercalated cells, type A intercalated cells. So in other words, the NH3 that we absorb is going to buffer the hydrogen ion, therefore improving the ability of type A intercalated cells to secrete hydrogen. So it will make it easier for you to secrete and buffer hydrogen. So it will allow you to cope with acidosis. So that's why when we have a mutation in this transporter, in this exchange channel here, which is the NHE4, we don't get the NH3, enough NH3 next to the collecting duct. If we don't get that, then we're not buffering the hydrogen ions. If we don't buffer the hydrogen ions, then the intercalated cells have less ability to secrete hydrogen. Remember, if we raise the hydrogen ion, the free hydrogen ion concentration in the collecting duct, that also inhibits the hydrogen pump. So we really want to capture that hydrogen ions in order for the hydrogen pump to keep working. So hopefully that explains to you why the mutation in NHE4 can cause metabolic acidosis. It's not important for the exam, but I answered it and I'll post it to you anyways. Have a great